Sapiomed Medical Podcast, Carbohydrate Metabolism. Episode 1, Fundamentals of Carbohydrate Metabolism. Hello, and welcome to the Sapiomed Medical Podcast, your go-to source for clear, concise, and clinically relevant content. I'm your host, Daniel Arhin Oduro, the creator and founder of Sapiomed, and today we begin our three-part series on carbohydrate metabolism, one of the cornerstones of clinical chemistry and human biochemistry. In this first episode, we're covering the fundamentals: what carbohydrates are, how they're digested and absorbed, the detailed steps of glycolysis, and how the body manages energy under both aerobic and anaerobic conditions. We'll also link everything to real clinical scenarios so you can apply this knowledge practically. Let's dive in. 1. What are carbohydrates? Carbohydrates are organic molecules composed of carbon, hydrogen, and oxygen, usually in a 1:2:1 ratio. They serve as a primary energy source for most living organisms. In humans, carbohydrates play critical roles not only in energy production, but also in cell signaling, structural integrity of cells, and more. They are especially vital for the brain, which relies heavily on glucose for fuel. Structurally, carbohydrates are classified into several categories. Monosaccharides. These are the simplest carbohydrates. Common examples include glucose, fructose, and galactose. These are typically absorbed directly into the bloodstream. Disaccharides. These are composed of two monosaccharide units. Examples include sucrose, glucose plus fructose, lactose, glucose plus galactose, and maltose, glucose plus glucose. Oligosaccharides consist of 3 to 10 monosaccharide units. They are less common in nature, but can be found in some vegetables and grains. Polysaccharides. These include starch and glycogen, which are storage forms of glucose in plants and animals, respectively. Cellulose, another polysaccharide, provides structural support in plants, but is indigestible by humans. In human physiology, glucose is the central carbohydrate. It is the reference sugar in terms of metabolism and the main molecule that fuels cellular respiration. 2. Digestion and absorption of carbohydrates. Carbohydrate digestion starts in the oral cavity, where salivary amylase begins breaking down starches into maltose and dextrins. However, due to limited time in the mouth and the acidic environment of the stomach, which inactivates salivary amylase, most digestion occurs in the small intestine. Here's how it works. In the duodenum, pancreatic amylase continues the breakdown of starch into disaccharides and oligosaccharides. At the brush border of the intestinal epithelial cells, enzymes like lactase, sucrase, maltase, and isomaltase cleave disaccharides into monosaccharides. Absorption. Glucose and galactose are absorbed via SGLT1, a sodium-glucose cotransporter. Fructose is absorbed via GLUT5, a facilitated diffusion transporter. All three monosaccharides enter the portal circulation through GLUT2 on the basilateral membrane of enterocytes and are delivered to the liver. The liver acts as a central processing unit. It can store glucose as glycogen, convert glucose to fatty acids for long-term storage, release glucose into circulation to maintain blood glucose levels. 3. Glycolysis, the central pathway of carbohydrate metabolism. Glycolysis is the most fundamental energy-yielding pathway. It takes place in the cytoplasm and is anaerobic, meaning it does not require oxygen. The glycolytic pathway converts one molecule of glucose, 6 carbon, into 2 molecules of pyruvate, 3 carbon. Along the way, it generates a small amount of ATP and NADH. Let's walk through the key steps. Investment phase, glucose. glucose 6-phosphate via hexokinase, or glucokinase in the liver. This step uses 1 ATP, glucose 6-phosphate, fructose 6-phosphate via phosphoglucose isomerase, fructose 6-phosphate, fructose 1,6 bisphosphate via phosphofructokinase 1, PFK1. This is the rate-limiting and highly regulated step. Another ATP is used. Cleavage phase 4. Fructose 1,6-bisphosphate is split into two 3-carbon molecules, glyceraldehyde 3-phosphate, G3P, and dihydroxyacetone phosphate, DHAP. 5-DHAP is converted into another G3P, so we now have two G3P molecules going forward. Payoff phase 6. Each G3P goes through several steps that yield one NADH and two ATP molecules. 7. The final product is pyruvate. Net yield per glucose molecule: 2 ATP, 4 made, 2 used. 2 NADH, 2 pyruvate. 4. Fate of pyruvate: aerobic versus anaerobic pathways. The fate of pyruvate depends on the availability of oxygen. In aerobic conditions, pyruvate is transported into the mitochondria. It is converted into acetyl coenzyme A by the enzyme pyruvate dehydrogenase. Acetyl-CoA enters the Krebs cycle, leading to further ATP production via oxidative phosphorylation. In anaerobic conditions, pyruvate is converted into lactate by lactate dehydrogenase. This occurs in tissues like skeletal muscle during intense exercise and in red blood cells, which lack mitochondria. Lactate can be transported to the liver via the Cori cycle, where it is converted back to glucose. 5th regulation of glycolysis. The glycolytic pathway is tightly regulated at 3 major steps: hexokinase/glucokinase, inhibited by G6P, feedback inhibition; phosphofructokinase 1, PFK1, activated by AMP and fructose 2,6-bisphosphate, inhibited by ATP and citrate; pyruvate kinase, activated by fructose 1,6-bisphosphate, inhibited by ATP and alanine. Hormonal regulation also plays a role. Insulin stimulates glycolysis. Glucagon inhibits glycolysis in the liver. 6th clinical correlations. Understanding carbohydrate metabolism is key in diagnosing and managing several medical conditions. Lactic acidosis occurs when lactate builds up faster than it can be metabolized, seen in conditions like sepsis, shock, cardiac arrest, or mitochondrial disorders. Symptoms include rapid breathing, confusion, and low pH in blood gas analysis. Enzyme deficiencies. Pyruvate kinase deficiency leads to hemolytic anemia due to reduced ATP in red blood cells. Phosphofructokinase deficiency, Tarui disease, a rare glycogen storage disease presenting with muscle weakness and cramps during exercise. Glucokinase mutations can cause a form of maturity-onset diabetes of the young, MODY. 7. Summary and clinical pearls. Let's wrap up with a quick recap. Carbohydrates are broken down into monosaccharides and absorbed in the small intestine. Glucose is the primary energy source, metabolized through glycolysis. Glycolysis is regulated and can proceed with or without oxygen. Pyruvate's fate depends on oxygen availability, aerobic metabolism for maximum energy, anaerobic when oxygen is limited. Clinical relevance includes conditions like lactic acidosis and rare enzyme deficiencies that impair carbohydrate utilization. Clinical pearl: always consider a patient's metabolic state, oxygen levels, organ function, and enzyme activity when interpreting abnormal glucose or lactate results. Thank you for joining us for episode 1 of the Carbohydrate Metabolism series. In our next episode, we'll explore glycogen metabolism, gluconeogenesis, and the hormonal regulation of carbohydrate pathways. Make sure to subscribe, leave a review, and share this episode with your fellow students and colleagues. You can find downloadable notes and references on our show page. Until next time, keep studying smart and stay curious.